Hi everyone, welcome to my channel Pure Biology. And today I come with after a long time with a new video. And why I didn't give any video for a long time, because I was busy for changing my setup and obviously in another place. So in that situation, I didn't give. I tried, but I didn't give any video for this problem. And but I will assure you that from today I give you the videos in every week. Okay, so continue with my channel. So this is the translation process. Here I will only describe about the involvement of the factors which are responsible for the translation process. Okay, and in my previous video I already have explained about the tRNA structure, so you just check out that one, and after this you continue with this video. Okay, so here the first topic is the ribosome, that is mainly the function of the ribosome is the protein synthesis, and this is consisting of the protein particles and small r RNA. Okay, and the ribosome, this has the two subunits. One is the large subunit, another is the small subunit, and you just see that each ribosome is made up of the two subunits, and generally it acts like a small migrating factors. Okay, it acts like a small migrating factor, and in E. coli, this is the example that they have the 30S subunit where 16S rRNA and 31 proteins there. Okay, but in case of 70, here I will obviously try to know or try to learn about the 70S subunit. It has the 3 sites. Okay, and this is very important for the translation process. The first site is the P site, second site is the A site, and the third site is the E site. The first site, P site, that is known as the peptidyl tRNA binding site. The second site, A site, that is known as the amino acid tRNA binding site. And the third one, E site, this is called the exit site. Now, what is the function of the P site, A site, and the E site? The P site, that holds the tRNA molecule that is linked to the growing end of the polypeptide chain. Okay. The second A site, its function is that to hold the incoming tRNA molecule that is charged with an amino acid. And E site is what? This is the exit site, because deacylated tRNA that exits via the E site. Okay, and now this is the second topic. Here I will try to explain with the activation of the amino acid and the attachment to the tRNA. First, the activation of the amino acid. What is the activation of the amino acid? The attachment of the amino acid to tRNA is the function of the group of enzymes called the amino acid tRNA synthetase. Okay, synthetases. That means the attachment of the amino acid with the tRNA, it is possible due to the presence of this amino acid tRNA synthetases. These enzymes are very important, and they form a covalent bond. Okay, they form a covalent bond between the tRNA and the obviously the amino acids. Okay, and when tRNA that is charged with amino acid, this corresponding to its anticodon, and it is called the amino acid tRNA. What is saying that when tRNA is charged with amino acid and corresponding, that means its anticodon site, and obviously you have to first know that which one is the anticodon site and the codon site. So you just check out the previous videos of transcription and tRNA structure. Okay, so it's clear. Just seeing or just learning about this tRNA structure, this topic is clear to you. Okay, and this what is saying that this when tRNA is charged with the amino acid corresponding to its anticodon, it is called the amino acid tRNA. Okay, and this charging process occurring in the two ways. First is that amino acids are activated by AMP forming and obviously forming of amino acid adenylate. This is the intermediate molecule. And the second step is that amino acid is transferred to the 3' end of tRNA. and there is a bond is formed between the COOH group of amino acid and OH group that is attached to the either 2' carbon or the 3' carbon of the sugar, and that is obviously present on the A site. Okay, so these are the two reactions. This is the first step, amino acid and ATP in presence of synthesis form the amino acid AMP and PN, and when they attach with the tRNA in presence of obviously synthesis, they form amino acid tRNA and AMP. Okay, so this is the actual process or involvement of the ribosome and the activation of the amino acid. I explained here, and it's very easy, and I explained here briefly, so I think you all understood. Okay, so I will try to explain the factors, that is the initiation factor, elongation factors. These are important for the translation process, so please you continue with my channel. Okay, thank you.